we see that in silico methods are there since many, many years now. And they are a standard tool to use to support our work in several steps of drug discovery and development. And I think they are there to stay in the years to come. So I think we shouldn't be afraid to use them, to look at them, and to be curious about them. So I think maybe the message is to be open to these new techniques and these new methods. And if they're helpful, then why not?
Welcome back to part 2 with Giuseppe Licari from Merck Healthcare, where we're tackling the toughest formulation challenges in biologics development. How do you predict aggregation before manufacturing? What can simulations really tell you about excipient protein interactions? And when do you know it's time to stop computing and start experimenting in the lab? Giuseppe shares practical workflows, real success stories, and honest limitations of computational approaches. Plus, he'll give you one actionable step to start incorporating these tools tomorrow. Let's jump back in. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brohlman, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics and market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. So we have done our homework. We showed that our molecule is developable. We did the formulatability. And now we're developing the proper formulation of the recombinant drug. So what are the specific in silico approaches you're using? Because it feels a bit to me like a puzzle, a very difficult puzzle to solve. You have so many different chemical components. You can have them all kinds of different concentrations, combinations of that. So how do you find the needle in the haystack?
Yeah, first of all, what is important to consider that for formulation development, you need to look at how the protein behaves in the environment along time. So you cannot really base your assessment just on a static picture of the antibody, but you need to look at the movie. And this is done in our field. Well, it's done in different ways, but the way I do generally is using a technique called molecular dynamics. And this is a field of computational chemistry where you can really see how molecules dance around, if you want to imagine it like this. So you really see how the protein moves and how its conformation changes over time. And then if you add your excipients, if you add your buffers, then you see how those elements will interact with your protein. Then you can also extract conclusion out of those interactions, because you can see really how those components might affect the stability of the protein, or how those components might change the properties of the protein. So I guess this is one of the critical points here, to look at the movie of the protein. And at the end, it's like doing an experiment, but in the computational field. So we really try to reproduce what we do in the lab. So taking your drug substance and putting your drug substance into a certain environment and looking at how it behaves over time. Of course, it's not exactly the same as we do in the lab, but it's a semi-realistic representation of the reality, and you can still extract interesting information and helpful information for your experiments.
And how do you go about real stability studies? Because this is what takes time and you cannot compress the time. Well, obviously, yes, you can do under stress conditions, but still it takes some time to confirm your outcomes. How do you combine the in silico there with the stability studies?
Of course, real-time stability studies, like to assign the shelf life, it's not something that can be directly computed or simulated in the in silico approaches, because one of the limitations of those approaches is that you can simulate only a short period of time. So you cannot simulate 6 months stability or so definitely this is out of scope of the in silico methods, but actually the stability of your protein in the long term is really tightly related to the properties of the molecule. So that's what we want to do. We want to study the properties of the molecule and understanding which properties are connected to the long-term stability of the molecule. Once you understand these connections, then you can try to tweak then the formulations to try to adjust the behavior of the molecule, and then you can somehow have an idea of the long-term stability, even if you are just simulating like for a short time. So that's somehow, I think, the strategy that should be followed.
And now looking ahead, because our industry, and I'd say in general, is evolving very rapidly with all kinds of technology. Let's name the buzzwords AI, machine learning, and so on. What is happening in the formulation development space? Where do you see this going to, let's say, in a few years? How are we going to develop formulations for recombinant proteins?
Yes, if you look at the AI space, so the machine learning is part of somehow of this field. So I think now there are several efforts to try to predict the formulation behavior of proteins using machine learning. So you need to have a really significant amount of data than to try to predict a certain behavior. So for example, you could try to predict a different formulation, like what could be the level of aggregates or the level of low molecular weights. But one of the issues right now is that we don't really have a lot of data to really build something that is robust enough to be applicable for several proteins and for several systems. So I think this is still one limitation of those techniques to be used in a field like formulation development. However, the AI now is helping a lot to really try to generate very good proteins. So in the field of discovery instead, we can use generative AI to really provide a protein that has like reduced amount of chemical liabilities, improved developabilities. So I think in the future, we will see more and more improvement of proteins early in the process. And that will have, I think, also a very good effect downstream. So in the formulation development or any other downstream step. So if you improve your protein already from the beginning, I think it will have a very significant impact later in the process.
Yeah, and I think that with the increasingly better generative AI models and also the much more powerful computational technologies or techniques, you will perhaps even be able to select the right sequence and then already predict what is the formulation of a molecule, if I think a bit futuristically.
Absolutely. Absolutely, yes. I'm really looking forward to the time where actually this in silico method will be able also to tell you this formulation is the right one. And then the experiments will be used just to confirm that the prediction was correct. So right now we are not really there still. We might need still to do some screening or some tests, but I think in a few years we might be able to just use experiments just to confirm the predictions. And that I think will have a huge advantage in terms of timelines, of reduction of time, reduction of costs, and so we can develop more and more molecules for the patients at the end.
To make our conversation very actionable, I would like now to look into how, for instance, someone who is working in a smaller company could apply that. Because the challenge is always, especially as we look in the future, it's exciting. We have amazing new technologies. You could do a lot of things. And I think in a larger company, that's also my experience. You're pretty fortunate to have a lot of resources. But when you're working in a startup company or in a small and mid-sized company, you have more limited resources. So what would be your advice to still be able to leverage some of the potential, at least, of these in silico approaches, even though you have a smaller budget or you have maybe not all these specialists in-house?
Yeah, sure. That could be of course, a problem for small companies or for a startup. Maybe the solution would be to do a small feasibility with an external provider, because now there are more and more companies that provide software as a service, for example. So you can somehow test those approaches through a third party and see if it gives some additional information or some valuable outcome for your project. So that's, I think, something that can be achieved by even smaller companies, because again, in silico methods generally are not so expensive from the computational side generally. So you don't need to maybe invest too much to test a few things. So maybe the advice would be to test with some external companies and see if it works. But of course, the best would be to hire a computational guy that will also really impact your internal knowledge. It's always better to have somebody internally. But yeah, we need to make compromise all the time.
Definitely. Absolutely. Before we wrap up, Giuseppe, what burning question haven't I asked that you are eager to share with our biotech scientists?
Well, maybe what comes next then in this field? This could be a burning question. Maybe the answer would be that I see now these new machines, new GPUs, new computational power that is increasing over and over. And I think in the future we'll be able to simulate bigger systems for a longer time. So I think simulations will be able to reproduce probably any step in development space. And this will really support more and more different phases that we are kind of having in the development field. So I think that with more and more computational power, we'll be able to do more and more. So I'm really looking forward for the years to come to see how this field evolves.
This has been great, Giuseppe. What is the most important takeaway from our conversation?
I guess that we see that in silico methods are there since many, many years now. And they are a standard tool to use to support our work in several steps of drug discovery and development. And I think they are there to stay in the years to come. So I think we shouldn't be afraid to use them, to look at them and to be curious about them. So I think maybe the message is to be open to these new techniques and these new methods. And if they're helpful, then why not?
Why not? Why not? Yes, scientists, why not? Why not use these technologies? Giuseppe, where can people get a hold of you?
Well, I guess we can exchange in LinkedIn. They can find me easily in LinkedIn if they search for my name. And I'm happy to exchange with anyone who is curious about those techniques.
Excellent. Smart biotech scientists, please reach out to Giuseppe, exchange on the in silico approaches. And once again, Giuseppe, it has been fantastic. Thank you so much for being on the show today.
Thanks to you, David, for what you do and for the invitation. And it was also a pleasure for me. Thank you.
What a masterclass in computational formulation development. Giuseppe has given us a roadmap from theory to practice, showing how in silico approaches are becoming indispensable tools in the biotech scientist's arsenal. If these insights resonated with you, take 30 seconds to leave a review on Apple Podcasts or wherever you're listening. Your feedback helps us bring more expert conversations to the biotech community. So thank you so much for leaving a review. And thank you so much for tuning in. Until next time, keep making bioprocessing smarter, one innovation at a time. All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.